Hello, and welcome back to Mammalian Anatomy at Keuka College. We're going to be continuing our lecture series on Chapter 12 and Part 2. In this video, we're going to be focusing on the neuron and the synapse. So, what is a synapse? A synapse is essentially where two neurons come together to communicate. So in this top diagram, what we can see is two neurons. Here is the axon of the first neuron communicating with the second neuron, specifically on its dendrites. These two cells will come very close together and will communicate with each other here. So we would call this kind of area a synapse. There can actually be two different types of synapses, an electrical synapse and a chemical synapse. So let's talk about these synapses and compare and contrast them. Electrical synapses are really interesting because the two neurons are directly communicating with each other using gap junctions. So they are connected to each other using these specialized protein structures that form tunnels between the two cells. And the action potential has the ability to directly cross over from one neuron to another. As a result of this, all electrical synapses are going to be excitatory. Excitatory stimuli means that it's going to ask the second neuron to get closer to having an action potential or stimulated to have an action potential. So it will always be kind of moving towards that direction. So excitatory says, yes, please, I want an action potential. Let's do it. These electrical synapses are pretty rare in the nervous system, and we'll talk about where they are located later on in the future. But one cool place that they are located that is outside the nervous system is the heart. The heart and the cardiac muscle cells are all connected using electrical synapses, and this is what allows the heart to be in coordination. And I'm such a geek about this. I can't wait to show you guys about the electrical signaling of the heart and how the heart beats. It's just going to be super cool. I can't wait. All right. But to recap electrical signals, the two neurons are connected by gap junctions. The action potential can travel directly across these gap junctions, and it will stimulate the second neuron to have an action potential. Over here, what we can see on this diagram is a chemical synapse, and this is what is typically found in the nervous system. One of the major features of chemical synapse is there's a gap between the two neurons, and this gap is called a synaptic cleft. Because the synaptic cleft is present, the action potential does not have the ability to cross from one neuron to another. So it needs to get creative and figure out a different way to communicate. And it's going to communicate using special chemical messengers called neurotransmitters. Neurotransmitters, for example, would be like GABA, serotonin, norepinephrine, those kind of things that you may have heard about in the past. So essentially what happens in a chemical synapse is that these neurotransmitters will be released from one neuron, and it will travel across the synaptic cleft and bind to receptors on the second neuron. These receptors will actually do signal transduction. It will send a secondary messenger into the second neuron to possibly generate a second action potential. Chemical synapses are really cool because the signal can either be excitatory or inhibitory. Excitatory saying to the second cell, yes, I want to have a second action potential. Can you get closer to the action potential and do this process? So it's a yes vote for the second neuron to have an action potential. Inhibitory action potentials, inhibitory signals, on the other hand, is going to tell the second neuron, no, I don't want you to have an action potential. I want it to actually make it more difficult for you to have an action potential. So it can actually ask the second neuron to remain quiet and not participate in communication. Both of these usually occur at the same time, and multiple neurons will connect to a single second neuron to actually vote for whether or not the second neuron will have another action potential. We'll talk about this process a lot more detail when we get to vertebrate physiology, but I just wanted to kind of give you an introduction to it here. So what I'd like you to be able to do is to compare and contrast chemical synapses and electrical synapses. How are they different and how are they the same? Let's go over the structure of a chemical synapse. So this would be more of a diagram on the exam. And so let's look at some of the few things that we can see here that are really interesting. So this whole kind of structure is a synapse. It is where one neuron is trying to communicate with a second neuron, and this will be specifically a chemical synapse. The first thing that we'll see is at the end of the first neuron, we'll see the terminal bouton, or what is also known as the axon terminal. I actually prefer the term axon terminal. And it's kind of a bulbous end. And within this axon terminal, we're going to have vesicles that are going to be filled with neurotransmitters. These vesicles are called synaptic vesicles, and they're going to contain different types of neurotransmitters. It could be dopamine, serotonin, whatever suits your fancy. And this is kind of where they're going to be stored until the nerve has to communicate with the second neuron. This kind of end of the axon terminal is known as the presynaptic membrane. It's called presynaptic because it comes before the synaptic cleft. And normally what would happen is when the first neuron is communicating with the second, the synaptic terminals will actually fuse with the presynaptic membrane and release the neurotransmitters into the synaptic cleft. This membrane over here, which is going to be part of the second neuron, is actually going to be called the postsynaptic membrane. The postsynaptic membrane should have receptors located here. So this diagram is a little bit bad. They need to add in receptors. And those receptors on the postsynaptic membrane are important because it will bind to the neurotransmitter and perform signal transduction, allowing a secondary messenger or another chemical to be produced inside the cell, leading to either the activation of an action potential or the inhibition of an action potential. So this would be a diagram that I would have on the exam, and I would ask you to label its parts. Thank you for listening to our discussion of synapses, and I will see you in the next video. Bye-bye.